Okay, welcome everybody to the Malaria Journal Club. Very happy today to introduce Tristan Dennis, who will be talking to us about tracing the invasion of Anopheles stephensi in Africa. Tristan is associated with the University of Queensland and also with LSTM. Has been working as part of the CEASE project, which is controlling emergent Anopheles stephensi in Ethiopia and Sudan. So over to you, Tristan.
Good. Thank you very much, Chris. Yeah, lovely to see some familiar faces in the audience, including my old group members and my mum. Yeah, so as Chris says, I'm a postdoc at the University of Queensland, but until recently I was also affiliated with LSTM, and my project was basically doing some genetic analysis to trace the invasion of Anopheles stephensi in Africa. So most of the audience are probably aware of Anopheles stephensi, but just a little bit of a recap. So Anopheles stephensi, which is this handsome mosquito on the left, everyone's been talking about it for the last decade or so because it's the everywhere malaria vector that's recently been recently, maybe 14 years ago, was discovered in Djibouti and has invaded a large swath of East Africa. And the reason a lot of people are worried about Anopheles stephensi is because unlike most of Africa's major malaria vectors, which live in rural environments, Anopheles stephensi is perfectly happy living in both rural environments and urban environments, like the ones we see at the top and the bottom left here. So it's perfectly happy living in sort of rural swamps, but it's also very happy living in construction sites and water tanks. And what that does is it raises the prospect of urban malaria transmission in Africa. So a modeling study, a couple of modeling studies by people like Marianne Sinker and Charlie Whitaker have shown that these are the sort of at-risk regions for Anopheles stephensi expansion in Africa. Potentially endangering around 126 million urban Africans with the possibility of year-round urban malaria transmission. So Anopheles stephensi was discovered in Djibouti City, in the sort of the crook of the Horn of Africa here. It's discovered in Djibouti City in 2012, and since has expanded to occupy a large portion of East Africa, including Sudan, Ethiopia, across the Red Sea into Yemen, and now more recently Kenya as well. So this is where we come in. Our part in investigating the invasion of Anopheles stephensi was the CEASE project, which ended at the end of 2025. But the CEASE project was a big sort of multi-center collaboration with partners from Ethiopia and Sudan, and a bunch of other institutions to work out what the sort of the ecology, the bionomics, and the vector competence of Anopheles stephensi was, to try and work out what the threat to malaria control that it might pose, to test different vector control approaches that might work apart from sort of bed nets and sprays, and also to understand in more detail the invasion and spread, as well as the mechanisms of resistance to the chemical insecticides that we normally use to control mosquitoes. So on the right here is the fantastic CEASE family at a meeting in Addis Ababa, and they've all played a massive role in getting this project over the line that was recently published in Science, and there have been a number of other fantastic outputs as well. So the three questions that we wanted to ask with our genetic analysis was, we wanted to work out where it came from, we wanted to work out how it spread around Africa, and we also wanted to work out what the genetic basis of insecticide resistance was, because if mosquitoes are resistant to insecticides, then the bed nets and sprays and the methods that we usually use to control them won't work. So these were really the three questions that I was hired to answer in my part of the project. So in terms of weighing competing theories about where stephensi comes from and how it gets around, really this topic has understandably generated a huge amount of discussion, but there's actually hitherto not really been that much data. So the previously published work, for obvious reasons, getting samples and genome sequencing them is difficult and expensive. So studies often tend to focus on specific regions within countries, often using relatively few samples. They tend to use short DNA barcodes, which are great for giving sort of species level insights, like working out what species the sample is, but not often not particularly informative for understanding, the really complex genetic histories that you get when you get a biological invasion. So we tried to take a more comprehensive view by sampling from across the native range and the invasive range. So on this map here, which is the map of where we collected or managed to get samples from, the shaded dark area is the native range of Anopheles stephensi. So it normally lives in India, South Asia, and around the Persian Gulf. So we managed to collect samples from, or get material from southern India, Afghanistan, and Pakistan, Iran, Saudi Arabia, and then in the invasive range in Yemen, Djibouti, Ethiopia, Kenya, and Sudan. And again, just a little bit of a recap of the invasion history. It was discovered in Djibouti in 2012, and then discovered in Ethiopia and Sudan in 2016, Somalia in 2019, and Kenya in 2023. So we collected around 600 samples from across the native and the invasive range, and whole genome sequenced them to sort of a 30x depth of coverage, to try and work out, where it comes from, how it gets around, and how it's resistant to insecticides. It's also just actually probably quite important to note here that there are some big gaps in our sampling as well. So, we weren't able to sample comprehensively across a lot of the native range. So there's a massive gap here between southern Iran and southern India. For a bunch of different reasons. Obviously collecting material from a lot of these places is really difficult, and also bureaucratic and regulatory barriers that prevent us from collecting material from these areas. But this also does limit our ability to be really, really definitive about matching samples from the native to the invasive range. However, using whole genome sequence data can give you insights into population histories over the finest spatial and temporal scales, and that's what we set out to do with our study. So question number one, where does it come from? So previously published mitochondrial work suggests that there's some similarity between the invasive population in Africa to South Asian populations. However, bearing the limitations of mitochondrial data in mind, we used our whole genome data to try and look a bit more definitively. There was a 2023 perspective suggesting that it could have been blown over by the wind from Saudi Arabia. Did it sort of sneak across the Red Sea from Saudi into East Africa? There was also a disputed detection in Egypt, and some have suggested that there's been this sort of cryptic population that's been living in Egypt and has expanded southward into Sudan. And people have also suggested, based on mitochondrial data, that there have been multiple introductions. So like all good genetic studies, we start with the PCA. And what this just shows us is how genetic variation is partitioned among our samples. And our samples group into three main clusters. We have samples from Asia, Afghanistan, Pakistan, and India. We have samples from the Middle East. So we have samples from Saudi Arabia and Iran. And then we have samples forming a big group over here from the Horn of Africa and Yemen. And we see a bit of substructuring in the Horn of Africa and Yemen on the right here, but we won't worry about that for now. We'll dig into that more a bit later. Again, when we're looking at the relatedness between all of our invasive and native range populations, I won't torment you guys with this figure for very long, because looking at a heat map on a presentation screen is a nightmare. But all we're trying to say with this figure really is that all the invasive range material, so these squares up here, a lower number indicates a higher level of relatedness. And what this figure is telling us is that all of the invasive range material is most closely related to itself. And then the most closely related external population to all of the African material is from Afghanistan and Pakistan. So, for example, here we see a very high level of differentiation between African and Saudi Arabian material. We see a relatively high level of differentiation between all of the Iranian and all of the African material. But again, all of the African material and the Yemeni material is very closely related to itself. And what this begins to tell us is that maybe the invasive population is derived from a single sort of genetic introduction event, and that introduction event might be most closely related to our sampled population in Afghanistan and Pakistan. Another way of looking at this is looking at what's called an admixture plot, and this tells us how, again, how genetic variation is partitioned among our samples. And are outlined in the red boxes all of the invasive range material. And the most important thing to take away from this figure is that the invasive range material all shares the same colors. So our samples from Djibouti, Ethiopia, southern Ethiopia, Kenya, Yemen, and Sudan are all to a lesser or greater extent derived from the same genetic pool compared to everything from Asia and the Middle East. While we have a little bit of a contribution from Asia in our African material. So again, all of our invasive range material looks like it's derived from the same invasive population, and that invasive population has some contribution and resembles to some extent the population in Afghanistan and Pakistan. Now, looking at a tree of population history, so this figure was produced by some colleagues of ours in working the team of Mike Fontaine at CNRS in France. And what this shows us is a tree of relationships between all of our populations. So the most important thing to look at, one of the most important things to look at is the horizontal aspect of the axis, the branch length. And what this tells us is if you look in the invasive range, so here we have Sudan, Kenya, southern Ethiopia, northern Ethiopia, Yemen, and Djibouti, is that the horizontal branch length separating all of these samples is very small, especially when we're looking at Djibouti and the node separating Sudan and Djibouti, which again suggests that all of the invasive range populations that seem to share a common ancestor on one branch are very closely related to each other. So we need to keep our sampling dates in mind. And with that in mind, we suspect that the fact that Djibouti isn't sitting at the most basal branch in this group is probably a consequence of the sampling dates. So all of these samples were collected in the invasive range between 2022 and 2023, which means that they've had time to evolve since they initially landed in the invasive range. So our hypothesis is that this basal group of nodes here probably corresponds to samples collected in Djibouti city, which then gave rise to a population in Yemen, and then gave rise to our population in northern Ethiopia, southern Ethiopia, and Kenya. Now, here we have a population in Sudan that sits off at the base of our clade-containing samples from Djibouti, Yemen, Ethiopia, and Kenya. And again, so this all shares a common ancestor with the entire invasive range. So what this tells us is either the Sudanese population is derived from the Djibouti population, but drift has made these branches look a little bit more separate, or the samples from Sudan are from a separate invasion event, but share the same invasive ancestor. So with these data, we can't tell the difference between those scenarios. But again, I think based on the sampling dates, I would wager that it's more likely that actually Djibouti is the ancestor of all of the invasive range population, which we'll go onto that in a bit more detail later. But what that would mean is the Djibouti population was the original landing pad for the invasion, which then seeded distinct invasive populations in Yemen, so that's this brown branch here, into northern Ethiopia, southern Ethiopia, and Kenya, that's these three branches here, and then another one off into Sudan up here. And we won't worry about the invasive range, the native range material from Iran, India, and Saudi for now. So yeah, again, we see a similar process reflected by the admixture graph, basically. So this contribution from India could reflect inconsistent sampling across the native range. So these are Afghanistan, Pakistan, and Indian samples. And because the admixture plot down here uses a similar, I might be butchering this a little bit, but uses a similar slightly related model to the one that was used to generate this tree, the fact that the admixture plot groups Afghanistan, Pakistan, and both Indian populations together, and then adds a small contribution to the Djibouti population in the invasive range, could have given rise to what looks like a possible Indian contribution to the original Djibouti invasion event. So yeah, in terms of where it comes from, our leading hypothesis based on our data is our similarity to our South Asian population. Whatever it is, it's extremely different to Saudi Arabia. We think the pattern in our data rules out the expansion of a cryptic population, and also the fact that it seems to be derived from a single invasive lineage effectively rules out the possibility of multiple introductions. So yeah, a little sidebar on multiple introductions. So I think people have suggested that there have been multiple introductions based on the presence of mitochondrial haplotype numbers. However, we don't think this is the case, and genetic diversity is actually on the whole extremely low. We'll get to that in a minute, but in general, compared to the nuclear genome, this is a phylogeny generated from nuclear genome markers, so the whole genome data, which splits all of our samples really neatly into the different geographic populations that we've defined. Compared to the nuclear genome, the whole mitochondrial genome doesn't really. So you can see here, compared to the nuclear genome, which incorporates millions of markers sampled from across all of our sample set and across the whole genome, the mitochondrial tree reflects that really poorly. So all the samples are all jumbled up. It's clearly not a very informative marker. It's able to reconstruct the broad difference between all of the invasive range populations, so the tan, blue, and green, compared to the native range populations in purple and orange. But really beyond that, it seems to be insufficient to resolve the invasion history in any real detail. So yeah, our hypothesis so far is that our samples share the greatest similarity with our material from South Asia. It all seems to derive from a single invasive lineage. We think that the most likely scenario is that Djibouti was the initial introduction point, and also that the mitochondrial barcode data that has been previously used to study the invasion is probably insufficient to resolve the invasion history in a huge amount of detail. So this comes to our next question then, is how has it gotten around? So people have suggested that wind might have moved it around. There's a lot of competing theories here. They're not mutually exclusive, but really, again, there's not really been a huge amount of evidence. There's been a lot of discussion about potential dispersal along transit routes, as well as just intergenerational spread. So that would just be, the mosquito moving under its own steam. So we're going to investigate this in a couple of different ways. So we're going to use we're going to rely on two concepts. We're going to rely on a concept called isolation by distance, and then we're also going to rely on genetic diversity. Isolation by distance, briefly put, I've screwed up my slide layout, but briefly put, isolation by distance is the concept that geography shapes genes. So I think it could be summarized really nicely by this 2008 figure here, which is a plot of genetic variation of human beings sampled from across Europe. So the map of Europe is in the top right, and the colors of the points are the samples from humans that have been collected and genome sequenced. And if you kind of squint at the map, you can see that the genetic variation of the samples is partitioned in a way that it reflects the geography from which they were sampled. So on the bottom left here, you have your samples from Spain and Portugal. Up here, you have your samples from France. Up here, you have samples from Ireland, the UK, Northern Europe, Central and Eastern Europe and Russia, and then Southeastern Europe and Italy here. And if you sort of squint, you can see that the genetic variation in these samples reconstructs that geographic sampling origin really nicely. And we can use that concept to study our invasion history and the invasion processes that have given rise to the observed pattern of genetic variation that we see in our data. So on the left here is what we would see if we had, for example, continuous, sort of a slow process of intergenerational dispersal where the organism moves across the landscape gradually. Your individuals here are these little blue dots. And on the bottom here is what you would see in a plot of genetic relationship versus geographic distance, as you would see as geographic distance increases, individuals become steadily less related, basically. So this is what you would see if there was a barrier separating the individuals that you had been sampling. So, on our plot of genetic versus geographic distance, you would see that genetic and geographic distance increase together for a little bit, and then there would be a big jump because your barrier would mean that there was a genetic break between your two sampled populations. And another thing we're interested in potentially looking at is long distance dispersal, for example, wind assisted movement or movement of groups of populations by human transport, and then you would sort of get these like low outliers on your isolation by distance plot. So by looking at the effect of the effective geographic distance on genetic distance, you can begin to make some inferences about what your invasion process might have been. So we are going to use isolation by distance and isolation by environment to work out where the mosquito moves. So we're going to examine the effect of geographic distance and environmental distance to work out how it gets around the landscape. So on the left here is a plot of estimated migration. So the points are where we have, the map is a map of the invaded regions. So this is Saudi Arabia, Sudan, Ethiopia, Eritrea, Somalia, and Kenya. The points are where we have sampled mosquitoes in the SEEDS project. So the gray points are where we tried to sample and we couldn't find any. The blue points are where we tried to sample mosquitoes and we found some. And the yellow points are where we tried to sample mosquitoes, we found some, and then we included them in this genome sequencing study. The grid and the color of the grid shows you the estimated migration based on the genetic data. So this uses a clever algorithm to estimate migration between your sampling locations based on your that relationship between genetic connectivity and geographic distance that I explained in my previous two slides. Where blue indicates a high level of migration and brown indicates a low level of migration. And again, this tells us a couple of things straight away. The first is that there is a massive barrier to migration between Saudi Arabia and the rest of the Horn of Africa and Yemen. It tells us there's a relatively high level of migration within Sudan. It tells us that there's a barrier to migration around Ethiopia here, in a region that seems to roughly correspond to the Ethiopian highlands, and also areas where we weren't able to find mosquitoes and where stephensi mosquitoes at least, are notably quite scarce. It tells us that there is migration occurring within this sort of a far region of Ethiopia, and then down into Southeast Ethiopia and Kenya, again with a sort of barrier to migration that seems to correspond to the highland regions in Ethiopia. And if you guys remember, that plot of isolation by distance where there was that big barrier, this is the plot here of genetic connectivity versus geographic distance within Ethiopia and Sudan. So we've grouped Ethiopia and Sudan into two distinct populations and calculated genetic versus geographic differentiation between them. And what we see here is that basically that genetic jump here that indicates that there is a barrier to gene flow between individuals of the different populations, where Sudan and Ethiopia and Kenya have been categorized as different populations. So we see a statistically significant barrier to gene flow between Ethiopia and Kenya and Sudan. Here, I've put it down here. And again, based on the admixture plot, the admixture tree as well, it's again a fairly nice illustration of the fact that the Sudanese invasion, Sudan down here, and the Ethiopian invasion, that's northern Ethiopia, southeastern Ethiopia, and Kenya, are geographically and genetically quite distinct within the invasive lineage. So while they share a common ancestor, they are then geographically quite separate. So yeah, based on our analysis of our genetic populations and also our landscape genetic analysis of isolation by distance, we can say that Sudan, Yemen, and Ethiopia, Kenya are three distinct invasion fronts. Okay, so within these invasion fronts, we want to know where did it originate from and how is it moving around. So again, lots of thoughts, but not really much hard evidence. People have suggested that it might have been human transport routes or expansion from cryptic populations. But here we're going to rely on genetic diversity. And again, PowerPoint has messed up my slide layout, so my apologies for that. But here we're basically going to look at genetic diversity as a memory of the invasion. So I think this is illustrated really nicely by the genetic history of human beings. So in Africa, where all the ancestors of all human beings, or anatomically modern humans, come from, you have the highest level of genetic diversity because humans lived in Africa for a very long time before they migrated out. And as humans have migrated out of Africa, they have successively lost genetic diversity. So you can see that as human populations have moved out into Europe, into Central Asia, Southeast Asia, and Australasia and America. In the Americas, you see this loss of these colored dots that indicates that as the migration and the invasion, biological invasion, I guess, has moved out of the origin point. It's successively lost genetic diversity. And in the context of our mosquito, we can use this to trace where within the invasive populations where it's been moving around. So because each successive population inherits only a sample of its source variation, and so diversity erodes step by step along the invasion path, we can read back along the route to work out, the entry point, because the highest genetic diversity will be in the point closest to the source. So here's a plot of, we're not going to worry about the bottom one for now, we're just going to focus on the top one of pi, which is a statistic of genetic diversity. Again, this is Afghanistan and Pakistan, and then our invasive populations in Africa and Yemen. Compared to the rest of the native range in Saudi Arabia and Asia, genetic diversity in Afghanistan and Pakistan is extremely high. And we think that's the reason why a lot of the, relative to a lot of the other external stephensi populations, most of the invasive range has a higher level of pi than its a lot of its native range correspondence. However, we think it inherited substantial diversity from the native range and has lost it successively as the invasion has progressed. So Djibouti here, which we hypothesize is the original landing point, and again, that's consistent with the history of discovery. Djibouti is the original landing point of stephensi in Africa, then into northern Ethiopia, southeastern Ethiopia, and then the darkest blue here is Kenya. So that is this stepwise reduction in diversity that's consistent with the invasion history and the history of discovery. And again, we have reductions in diversity in Sudan and Yemen as well. So the diversity tracks the invasion and discovery history with substantial amounts of diversity inherited from the native range that has then been lost in stepwise introductions as the invasion has progressed throughout Africa. Again, so we have a little zoom in on the Afghanistan and Pakistan and the invasive range here, showing that nice stepwise reduction. So that's consistent again with the history of discovery, because Kenya was the most recently discovered population. This is a statistic called homozygosity, and homozygosity will tell us how recent and how severe the genetic bottlenecks that accompany invasions have been. So, when populations invade, they undergo these massive population size reductions, and this leaves footprints in the genome. And what it leads to is an increase in the number and fraction of an individual genome that sits in long runs of homozygosity. So these are long tracts of homozygous genotypes that are inherited when related individuals breed with one another. So we would call it inbreeding or consanguinity. And what we see in the invasive range, again, is that compared to the native range, which are these samples here, we see a marked increase in the fraction of the individual genome in runs of homozygosity, which indicates a massive excess of recent inbreeding. So in Djibouti, that effect is the smallest, because Djibouti is the oldest population in the invasive range and has had a long enough time to build up that diversity back again. But then when we go into northern Ethiopia, which are these teal samples here, southeastern Ethiopia, which are the light blue ones, and then into Kenya, you can see that the number and fraction of the individual samples in runs of homozygosity begins to increase. In Sudan, we have this massive inbreeding effect where we have this big smear of samples off to the right of our plot here, indicating substantial amounts of the genome are now in tracks of homozygosity. So for contrast, an individual FROH of 0.6 is really high. So in terms of domestic pets, for example, that people like to inbreed, 0.4 would be about the inbreeding level of a pug or a beagle or an English bulldog. So what we're seeing is, as a result of this biological invasion, our mosquitoes have lost a substantial amount of diversity and are undergoing severe genetic bottlenecks. So again, the inbreeding seems to track this history of discovery, whereby more recent discoveries are more inbred. So this is some more work that's been done by our colleagues at CNRS in France, and what they've looked at is what's called genetic bottlenecks, where these y-axis are estimates of genetic population size in recent time. So on the x-axis is time in generations. And what we can see is that compared to the invasive range in Afghanistan and Pakistan, all of our invasive range populations in Djibouti, northern Ethiopia, southeastern Ethiopia, Kenya, Sudan, and Yemen have all undergone these big reductions in population size, these big genetic bottlenecks, and all have began to undergo some level of discovery. So again, this tracks the discovery history. So Djibouti undergoes this bottleneck first, northern Ethiopia, and then southeastern Ethiopia. Kenya more recently seems to have undergone these like recurrent bottlenecks and hasn't recovered as much. Sudan and Yemen as well. So in terms of time, it's difficult to pinpoint an exact time. This is in generations. And there are a lot of assumptions that we make when we do these kind of analyses about things like mutation and recombination rate. So we have to be cautious about interpreting too literally what the generations means. But with a generation time of 11 per year, this would hit roughly around the time of discovery of stephensi in Djibouti, and then tracking that discovery history across the invasive range, which is really neat. So we can look at, right now, all of our analysis has been done on individuals that have been grouped into genetic populations across the native and the invasive range. But we can look at this in much finer detail by examining genetic diversity in each sampling location. So, one question we have is, where did it originate? And how has it sort of moved across the landscape? In addition to our migration analysis, where we see that there's been this barrier to migration in the Ethiopian highlands, and that Sudan, Ethiopia, Kenya are distinct invasive propagules or invasion fronts. We've plotted genetic diversity on each sampling location. So what we see is that, where red is high and green is low and blue is even lower, where we see that there's substantial amount of diversity in Djibouti, which we've seen before, more in the sort of Afar region of Ethiopia, and then decreasingly less as we move into southeastern Ethiopia and Kenya. Sudan is much less genetically diverse than northern and central Ethiopia generally. And again, we see this sort of general trend towards decreasing genetic diversity as we move across the country. And so one question we wanted to ask was, does genetic diversity decrease with distance from the supposed port of introduction? So we think stephensi arrived in Port Sudan in Sudan. It was first discovered there, and then was first discovered in Port of Djibouti for the Ethiopian and Kenyan invasion front. And so we can plot this. So this is pi, and this is runs of homozygosity or inbreeding for the Ethiopian incursion and the Sudanese incursion over distance to the port servicing that country. So the main port servicing Sudan, and the Sudanese invasion would be Port Sudan, which is also where it was first discovered, and Port of Djibouti for Djibouti, which is also where it was first discovered. So these are the suspected ports of entry, and again, these data coincide broadly with the history of discovery, and they sort of order and direct the spread. So genetic diversity in Ethiopia and Sudan falls, and runs of homozygosity or inbreeding rises with distance from the port in a stepping stone sequence outwards, which also, well, more on that in a second. And the shape of this diversity distance relationship tells us how each front moved. So in Ethiopia, you have a much steeper curve, where you have this more sort of incremental crawl, where the invasion is moving more slowly and it's losing diversity, but it's also recovering diversity with time. Whereas in Sudan, you have a much flatter decline of genetic diversity over distance, which suggests that this has been a much more rapid, genetically homogeneous sweep across the country. And one thing that this correlates with really nicely is the migration analysis that showed that there were many more barriers to migration across Ethiopia and Kenya than there were in Sudan, where there was this relatively unrestricted corridor for migration across the country. And we see this reflected not only in the genetic connectivity and isolation by distance data, but we can also see it in the genetic diversity data as well. Compared to distance from the nearest coastline, so it's been suggested, that maybe, the stephensi has expanded from unsampled locations in Somalia, and obviously, while we can't rule that out, we think that our statistical model, which favors port versus coast, so the lines on each point are, the genetic and geographic distance from each port, and shown in these plots here, we haven't got the plot for coast, but we did the same analysis, but for the coast, saying, is distance from the coast a better explainer of the observed decay in genetic diversity than distance from the port? And what we find is, no, it's not. So it's the ports. So yeah, genetic diversity and bottlenecks. So genetic diversity radiates out from the suspected ports of entry for the Sudanese and Ethiopian, Kenyan invasions. And the timing of the genetic bottlenecks and the sort of the general pattern of genetic diversity is consistent with the invasion discovery. So this argues against other invasion propagules that have originated in Somalia, although again, we can't say for sure. And it also argues against the idea that there are cryptic populations that emerged from Egypt or elsewhere, because the timing of our population crashes is consistent with the history of discovery. So if we plot it on a map, this is what our hypothesis is. So we have an invasion so far. So based on the admixture plots, the genetic differentiation plots, the heat map that I showed you guys in an earlier slide, that confusing looking tree, we think that, again, consistent with the history of discovery, Djibouti was the landing site. It was the place where the invasion arrived in the region. And then somehow it spread up the Red Sea into Sudan, either from the same origin as the Djibouti population or seeded by the invasion in Djibouti. And it also jumped across the Bab el-Mandeb Strait into Yemen. And from Djibouti, it radiated outwards into the Afar Triangle, where it was stopped by the geographic barrier of the Ethiopian highlands. And it then spread out and around into southeastern Ethiopia, across into Somalia, and then down into Kenya. Whereas in Sudan, faced with much fewer geographic barriers, it spread much more quickly and much more genetically homogeneously across the country. So again, why is this? I mean, I think, the migration analysis showed quite nicely that the terrain might be playing a role. The highlands might be playing a role in slowing the invasion and constraining the spread into Ethiopia and Kenya. But we want to look at this in more detail. And just, I mean, even from a distance perspective, I can't remember the distances off the top of my head, but the invasion in Sudan has moved much more quickly in a much shorter period of time in terms of mosquito discovery than the invasion into Ethiopia and Kenya, which you have from 2012 to 2022 when it was discovered in Kenya and Sudan. I think it was discovered all over the country in between 2016 and I think 2018. I think that's right. So yeah, why is this? We want to look at the landscape in a little bit more detail. And again, we're sort of comparing and contrasting our migration analysis and our genetic diversity analysis, and we want to use a more formalized landscape genetics approach. So yeah, my question is basically, were the mountains a barrier for invasion? Because based on our sort of correlative migration analysis, it looks like that might be the case, but we need to develop a more robust statistical framework for actually testing this. So what we did is we downloaded terrain data, like satellite data, for elevation. This is a plot of elevation across the region. And it's also just worth pointing out how beautiful the landscape is here. It's really an incredible landscape and a really beautiful topography in Ethiopia and Sudan. But so we download satellite data for elevation, humidity, temperature, rainfall, human travel time, wind travel time across different grid cells, and human population density. And then we calculate what are called resistance distances between our sampling points. So this is an example of what the raw data look like, which is basically where the algorithm looks for every single possible path between your sampling locations across each different terrain type. And then you basically build what's called a Bayesian regression model of all of our different resistance types, and work out the extent to which each landscape type contributes to the observed pattern of genetic variation. Now, on the top left here, we have a plot of correlation between our different landscape covariates. This is humidity, vegetation, human transit friction, geographic distance as a null model, because it's important to control for the effective spatial autocorrelation, rainfall, elevation, human population density, mean wind travel time, and genetic variation. And we can correlation between all these different covariates, because in a statistical modeling analysis, you want to make sure that you're excluding all of your correlated covariates. But what it basically looks like is that everything is really highly correlated, which makes total sense, right? Like, of course, increase in elevation is correlated with an increase in rainfall, which is also correlated with an increase in the time it takes for a human to cross a pixel, which is also correlated with the inverse of population density. So what this says straight away is that actually defining specific landscape characteristics that might play a role in shaping mosquito dispersal is going to be statistically very difficult. But we tried anyway. And what we find is that this is a plot of model performance loss, where when you remove a variable, you compute the amount of performance that your model loses by losing that variable. And what we find basically is that mean wind travel time, and once we remove friction, because it's very well correlated with the other variables, mean wind travel time and elevation seem to emerge as the strongest variables shaping genetic connectivity among our sampled mosquitoes. And this is the plot of our model simulations and our model performance, showing that although it's not a perfect fit, it's a fairly reasonable one. So again, it's very hard to say exactly what landscape features are driving or shaping mosquito dispersal. But again, a combination of intuition and interpreting our migration analysis, and also looking at the outputs of the model, that it would make sense that elevation and topography play a massive role in shaping the movement of the mosquito, and that plays into why a lot of the variables were correlated with one another. But what we see is that this is the plot of wind travel time across the map, and this is the plot of elevation across the map. And so we think that both wind and topography correlated with all of these other factors, like it being colder because mosquitoes don't like the cold, it being more difficult to move around in general, might be the factors that have shaped why it moved much more slowly throughout Ethiopia and into Kenya than it did into the Sudan, which is hot, flat, and windy. So yeah, what we see, 3 invasion fronts radiating out from the ports, and topology and wind are the factors that seem to be influencing invasion spread. So final question, the architecture of resistance. What is the genetic basis that underlies pretty widespread resistance to pyrethroids and other vector control insecticides across the invasive range? So we do the time-honored genome-wide selection scan, the bread and butter of the group. These are plots of the selection statistic H12 across each of our 3 mosquito chromosomes. We have chromosomes 2, 3, and X. Each row is a genetic population, starting off with Saudi Arabia and Iran up here, Afghanistan and Pakistan, Djibouti, Ethiopia, Sudan, and Yemen. And we see a couple of things straight away. We see that when we go further into the invasive range, it becomes very difficult to pick out a clean signal. And that's because the selection statistic that we use detects localized reductions in genetic diversity. Now, if you remember from my earlier slides, we've already had substantial genome-wide reductions in genetic diversity that mean getting a clean signal across the genome is going to be quite difficult, because a lot of the genome has reduced genetic diversity. The second thing we see is we don't see any selection at target site resistance loci like the VGSC, which play a massive role in resistance in other major malaria vector species like Anopheles gambiae and Anopheles collitzii. But what we do see is selection across the native and the invasive range of the metabolic resistance determinants like the CYP6 family, the GSTE, and the carboxylesterases here. And we see that there seems to be what looks like a distinct resistance profile in both the native between Saudi Arabia and Iran, and the invasive range in Afghanistan and Pakistan and Africa. We can look at this a bit more closely by looking at the pattern of haplotype clustering. So what we would expect to see in the presence of strong selection that would indicate involvement in insecticide resistance is what we get on the right-hand side here, which is where you have these big clusters of identical haplotypes shared between invasive range populations here, which are these colors. By contrast, in the voltage-gated sodium channel, the VGSC, we don't see any evidence of strong selection. And again, what we see is selection at the GSTE, the cytochrome P450 subfamily 6, the carboxylesterases, these metabolic resistance determinants that confer resistance by metabolizing the insecticide rather than mutations in the protein channels that the insecticide binds to. And what we can also see is that we see haplotype sharing, these orange haplotype sharing between Asia and the invasive range in Africa. So here, for example, we see an identical CYP6 haplotype between Afghanistan and Pakistan, northern Ethiopia, Sudan, Kenya, southern Ethiopia. So this tells us two things. It is unlikely to be target site resistance. It looks like it's all in the metabolics, and it also looks like insecticide resistance was also introduced from the native range in Asia. So the mosquito arrived in Africa pre-armed against the insecticides used to control it. Now, this is a plot of copy number variations. So at the metabolic resistance loci, these CYP6s and GSTs and carboxylesterases, the way the mosquito is able to become more resistant is by expressing more of these proteins, and the best way to express more of these proteins is to produce more of them. So in the genome, you get this copy number increase at the metabolic resistance loci. And this is a heat map of copy number variation at these metabolic gene families, carboxylesterase, CYP6, and GSTE in the native range, Saudi Arabia, Iran, Afghanistan, and Pakistan, and India, and then in the invasive range in Africa, so Ethiopia, Djibouti, Ethiopia, Kenya, Sudan, and Yemen. And we can see that the genetic basis of this copy number variation is remarkably uniform across all across the three metabolic families, across the entire invasive range. And it's shared really with Afghanistan and Pakistan, whereas the profile of copy number variation appears to be fairly distinct compared to Saudi Arabia and Iran. And again, this is consistent with our hypothesis that the invasion originated in Asia, and the basis of resistance was introduced from Asia as well. So that copy number variant profile at the metabolics was introduced from Asia during the invasion. So yeah, to sum up, our data suggests that, again, consistent with the mitochondrial markers, but I think we show nicely that it's probably an Asian origin. Obviously, we have gaps in our sampling, so we don't have that smoking gun. But it shows a remarkable similarity in Africa to the samples we collected in Afghanistan and Pakistan. We think our data favor an initial establishment in Djibouti, consistent with the history of discovery, that seeded 3 invasion fronts in Ethiopia, Kenya, Sudan, and Yemen. These were separated by the Ethiopian highlands and the Red Sea. And the spread of this invasion and the contrasting speed at which the invasion has been able to develop in Sudan and Ethiopia, Kenya has been strongly influenced by the landscape. So again, thank you all so much for listening. That's the end of my talk. I just want to say again, just a massive thank you to all of our fantastic SEAS family in Sudan, Ethiopia, and at LSTM, and then all of our wonderful partners who contributed materials from all over the world and helped out with the analysis and moral support and all that. It's been a tremendous pleasure to work with you all, and we're all just delighted that the project has landed somewhere like Science. So couldn't have done it without everyone, and it's been great to work together. So yeah, thank you all for listening, and I'll take any questions that you have.
Thanks, Tristan. That was awesome. What an amazing piece of work. Thanks for taking us through that. I think we should be able to share a link with the science paper, which I recommend that folk go and check out. It's really cool. I have many questions for Tristan, but I would like to ask the audience. And I don't now know how I do that because Teams has updated and there's a new interface. Right, so I've got hands. I see Raoul has his hand up. So I'm going to unmute. We'll see how that works. How do we do this? Could I ask that questions are put into the chat? Maybe if I make Raoul a presenter. Okay, Raoul, do you have a question?
Yeah, thank you, Chris. Finally, you did it. Congratulations, Tristan, for this wonderful analysis of work. Can you hear me?
Yeah,
yep.
I can hear you fine.
Okay, so it's true. I have a couple of questions, but I will just ask 2, and then the other maybe we can go by email. So at the end of the day, you use many analysis to see what is the origin of the invasive species Anopheles stephensi, and also how the theory of its spread, especially in East Africa. But at the end of the day, because I went through the paper, it's a lot of hypotheses. And then my question is, what kind of analysis could we really use to answer that question, to say that is the origin, and then that's the way the invasive species spread? Because all the tools or the analysis you use will still end up with hypotheses.
And I think that's probably all these kinds of data will give you in the end, right? Because your genetic data are just a reflection, a lens of looking at the history of your population, and the history of the invasion, because you can't directly observe, or it's very difficult to directly observe things like, intergenerational movement in mosquitoes, and we haven't been able to identify specific ships that the mosquitoes arrive from. So I think, our data give us what we think is the best hypothesis yet, or the most conclusive hypothesis yet. But like all hypotheses, if someone comes along with more data and more conclusive analyses, then we'll say that's a better job. But I think, looking at things like what would give us a more definitive picture or a clearer scenario of the invasion and its origin, I think obviously, sampling more extensively across the native range would give you much more of a clear view of where it might have come from. I think, given our data, a likely origin point might have been southern or coastal Pakistan or northern India. But again, securing material from Pakistan and India, the regulatory barriers are extremely high. So it's very difficult to collaborate in these environments. And again, sampling across the invasive range to identify what the landscape determinants to spread are, sampling more comprehensively. But again, you run into difficulties collecting samples quite quickly when you're working in these environments. So I think, like everything, more samples and more data. But I think, again, just bearing in mind that these genetic analyses are just a lens through which we can view processes like dispersal and organism origin. But I hope that goes some way to answering your question.
Thanks, Raoul. Thanks, Tristan. So we've got some questions in the Q&A. I'll read these out so folk can hear them. So Henry says, thanks, Tristan, for the great talk. Do you think the reduced diversity of stephensi populations in Africa may make the species easier to control on the continent? Or do you think the demonstrated selection at resistance loci will overcome this?
Yeah, it's funny, isn't it? I mean, people talk about things like mutational load and the accumulation of deleterious mutations and the impact of fitness that might have. I think in practice it would be quite hard to prove, and it certainly doesn't seem to have slowed it down. So my guess would be probably not. It seems to have done very well for itself, and it's already carved out like a very large chunk of the continent for itself. So I would say, to your first question, probably not. I think, does selection in the invasive range make it harder to control? Again, obviously selection at the resistance loci, there's this like uniform resistance basis. So I think, in theory, you could say that, if you understand geographically where your resistance determinants are located, you could then target populations with specific insecticide formulations. And again, it looks like the resistance basis is pretty uniform. So if it was possible to target insecticides operationally, that would be difficult because everything looks the same. So, the same insecticides will fail in Ethiopia as they will fail in Djibouti, because that resistance basis is so homogeneous, I think. And then in terms of like adaptation to its new environment, we're working on it at the moment to see if there's any, to try and disentangle the signatures of genetic bottlenecking and selection, basically, because of that noisy profile that we see in the invasive range. It's quite hard to tell.
Thanks, Henry. Thanks, Tristan. Just to hijack very briefly, with this big sort of wave of invasion across the African continent, and the big fears about the impact that this will have, particularly on urban transmission, do we have any evidence of that yet, where we're seeing sort of concomitant rises of urban malaria with this kind of invasion?
I think there are definitely people in the chat who would be much better qualified to talk about the actual risk. There have been two published case control studies showing the presence of stephensi has been linked to outbreaks of malaria in Ethiopian urban environments, and then there's the sort of the correlative data from Djibouti showing that stephensi arrived when malaria was under control in Djibouti, and then it arrived, and then there's been this sort of big uptick in malaria cases in Djibouti. But yeah, I guess it's quite hard to say because its range doesn't seem to have expanded to include like a massive urban area. So yeah, I think we're sort of 14 years on, and I guess the jury might still be out as to how apocalyptic some of the predictions might have been. But yeah, I'll leave that to those in the chat that are more qualified to talk about that than I am, the epidemiology of it.
Thanks, Tristan. You've got a couple of minutes left. There's a question from Kelly about trees. Thanks for an interesting talk. Could I ask how you made the tree? Did you apply a molecular clock? If you did this with a relaxed clock, even if the substitution rate was not accurate, it could give you relative divergence times, and you could account for sampling times using such methods. Were the clades you observed for each geographic region monophyletic?
The clades, so it was a genome-wide neighbor joining tree, so we didn't apply for the circular ones, we didn't apply a molecular clock or any kind of substitution model. All the geographic clades were monophyletic. The admixture tree is basically a tree of the covariation in different allele frequencies. So again, there's not a molecular clock or a substitution model as such. With respect to divergence time, we have been playing around a lot, not with phylogenetics, but with using ancestral recombination graphs to look at time to most recent common ancestor in different geographic groups, and we haven't started using them in stephensi yet. But my work in some Pacific vectors that I'm working on suggests that really resolving decade level events is going to be really difficult. So I would be surprised, although obviously delighted, if such approaches were able to resolve those events in such fine-grained detail. I think, but then again, the diversity data and the effective population size data seems to resolve it quite nicely. So yeah, who knows?
Unfortunately, we've run out of time. I think we could probably sit here and discuss this for quite a while. So just to say thanks again to Tristan. Yeah, super cool. Everyone should check out the paper. I think the link has been provided in the chat. And yeah, thanks to everyone that came and listened and asked questions.
Thanks for having me, everyone.
Catch you at the next one.